Welcome, everyone. We made it to the end. We made it to the 3MT Awards Ceremony. And as always, we are starting with a keynote talk this time around from Thomas Vogl, who is a PI at the Medical University of Vienna, more specifically at the Center for Cancer Research. I truly believe he's like the embodiment of a STEM researcher, combining science, technology, mathematics, and as well as engineering. And I hope you enjoy the talk as much as I enjoy his work, which is about deciphering the human antibody repertoire against the microbiome in health and disease. So Thomas, thank you for joining us. Thank you for taking the time. I know you're very busy and the floor is yours.
Great. Thank you very much for the introduction, Kais, also for the invitation to present here. So I think it's always nice to present one's work, and I think this is like a great framework for that. Yeah, so basically you have already introduced the title. You've already said a few words about me, but maybe just to extend on that a little bit, just where I'm from and what my training is. Originally, I'm from Austria, and I studied molecular biology in the undergrads and PhD, so like really hands-on work in the lab, wet lab experiments. But then I realized nowadays we're having larger and larger data sets. So I was more interested in going into the direction of bioinformatics, data science, learning how to work with such large data sets using machine learning. So I did postdocs in Australia and Israel, which were more on the data science side and technology development. And since about 2 years, I'm a group leader here in Vienna at the Medical University. And we're doing a combination of those two things. On the one hand, we use liquid handling robots to generate a lot of data in the wet lab. And then we use these bioinformatics and machine learning approaches to interpret that data. And what we work on biologically, the questions that we're interested in are the human microbiome. So all these bacteria that we have in our body and how they influence different aspects of human health, especially how they interact with our immune system. And the really striking thing is that humans are covered in as many microbial cells as we have body cells. So statistically, for every body cell, you have a bacterial cell. Obviously, those bacteria are much smaller than the human cells, but still they account for about 0.5 kilogram of biomass that you carry around. And most of those microbes are in our gut, and they influence many different aspects of human health, how we metabolize food, also the onset of colorectal cancer or the response to certain cancer treatments, as well as the onset of autoimmune diseases. And you can see all of these papers, they were just published in the last decade. So this is like a field that's quite new, simply because, let's say 20 years ago, it had not even been possible to study this diverse gut microbiome because sequencing technologies were not, powerful enough, and you could not get a high enough throughput to cover all of those microbes. And nowadays we start to appreciate how important these microbes are for our health. And it's not merely the presence, that they are in our gut and that they're making certain metabolites. It's also how they modulate our immune system, especially how they interact with our adaptive immune system. And why is that necessary? Why do we need to make immune responses against these harmless commensal bacteria? Well, I've already mentioned that we have large quantities of them in our gut. And now imagine if you have a small injury on your skin, like a small scratch, and you get some environmental bacteria in there. This can easily lead to an infection, inflammation, sepsis, and you can die from that. Before we had antibiotics, people would regularly die just from garden work, like a small injury, it gets infected, and there's little you can do. And those environmental bugs, they're in tiny quantities, micrograms, I guess. And now imagine you're carrying 0.5 kilogram of bacteria in your gut, and your gut is an especially delicate environment, right? It needs to be semi-permeable so you can take up nutrients from the food that you're eating. So if you have a small injury there, you will have a lot of bacteria leaking into your bloodstream, getting systemic exposure, which can then also lead to sepsis and ultimately death. And why is that not happening? Because our body invests a lot of energy into our immune system, and we're producing large quantities of antibodies, IgA in the gut and IgG in the blood, that are targeting those microbes and that keep them from exerting any harmful effects. And even though we know that we make these large quantities of antibodies, we don't really know what they do. We don't know which exact species they are recognizing. We don't know the exact antigens, the epitopes, the structures that these antibodies are targeting. And we also don't know how these immune responses are changing mechanistically between health and disease. And the main reason that we don't have answers to these questions is simply that conventional methods like ELISAs or peptide arrays only allowed to study hundreds or maybe thousands of antigens. So with an ELISA, because you all know that if you're, for example, testing if you've been exposed to coronaviruses, you would take one protein, you would take a spike protein from the coronavirus, put it on the plate, on a plastic plate, and see if you have antibody binding against that. But obviously the microbiome is not represented by one protein, it's rather multiple proteins from multiple bacterial species. So you can use peptide arrays where you can spot thousands of proteins on a glass slide and you can measure them in parallel. But still you need to make each and every one of those proteins in beforehand, which is quite laborious. And even if you can do thousands of proteins, that's not enough to cover the microbiome, because these bacteria that we carry around, they come from basically thousands of species. Every species has thousands of proteins. So it's more a space of 100,000s to millions of structures of molecules that could be recognized. So we need methods that scale by orders of magnitude beyond these conventional approaches. And that's basically what my lab is doing. We're using a high throughput method where we are combining synthetic DNA libraries with phage display and next generation sequencing. And thereby we can really study 100,000s of antigens in parallel. And the way that this works is first we start out by sequencing the microbiome, so we know which genes are present there. And then we annotate proteins, but we don't make each and every one of those proteins separately, which would be very expensive. Much rather, we reverse translate the amino acid sequences to DNA sequences. And these DNA sequences we can order on commercial microarray slides. So we basically get our antigens, our microbiota proteins encoded, 100,000s of variants at low cost. And then we take this DNA and we clone it inside of bacteriophages in such a way that every phage makes a different protein on the surface. So we have a phage library that represents all of those microbiota antigens. And if you then have an antibody that will bind the antigen in the gut, it would also bind the antigen on the phage. So you just mix these phages, this phage library with human blood, and then you will get binding events, and eventually you immunoprecipitate the bound phages, so you wash away the unbound phages. And eventually, how do you know which antigens are bound? Well, every phage carries the DNA encoding the protein on the surface within itself. So if we sequence these phages by Illumina sequencing, we will get higher read numbers for enriched phages. And this is shown here for 2 antigens, but it works for hundreds of thousands of antigens in parallel. And this approach is called phage display immunoprecipitation sequencing, PhIP-seq. This has been pioneered by Steve Allerger's lab at the Broad Institute of MIT and Harvard. So they have mostly used this for studying viruses and the human proteomes, so self-reactivity, autoantibodies, and we were the first to adapt this for the microbiome. And one key aspect of this technology is the library, so basically the antigens that you're encoding, because you can only ever detect things that are part of this library. So our current library is made of about 244,000 variants. About half of them come from healthy individuals. So we sequenced the microbiome of 1,000 healthy individuals, and we picked antigens from there. But on top of that, we also have probiotic strains, pathogenic strains, databases of virulent factors, toxins, and so on. And if we then take one blood sample of a healthy person, which sort of signal do we get? We get thousands of different signals, and many of them come from obvious controls like Epstein-Barr virus or rhinovirus. These are things that 80 or 90% of the population are exposed to. We also have a higher signal density against, these toxins and virulence factors, such as coming from Streptococcus and Staphylococcus. But the really interesting part is that this whole group of commensal gut microbiota and even probiotics also elicits very strong responses. And these are even harmless bacteria, such as lactobacillus. So this shows that humans make systemic antibody responses against their own gut microbiome. And that's just in one person. So the next question is, does everybody have the same immune responses or are they different? So next we started using our liquid handling robots where we can measure thousands of samples to analyze the immune repertoire of about 1,000 healthy individuals for whom we had a lot of metadata. So we have age, sex, microbiome composition, and different blood tests for them. And then we just check, does everybody have the same antigens? Do they have different ones? And that's summarized in this histogram here. So basically you have the number of individuals from 1 to 1,000, and then you have how many antigens appear in how many individuals. So you can see there are many, many antigens, more than 100,000 that appear in less than 10 individuals. So this is a very unique, this is a very, very personalized, a very private immune response. But then on the other hand, you also have universal immune responses where 100% of individuals recognize exactly the same antigen. And then there is everything in between, things appearing in 20, 40, 60% of the population. And then the next question is, what does it mean if some people have these antibodies, others don't? So next we were looking for associations with human health. And we have shown that there are, for example, strong associations with age and sex. So basically in older individuals, you have very different antibody repertoires than in younger individuals. What you see in these plots here is that every dot is a peptide, and then you have the antibody binding in the youngest 20% of our cohort or in the oldest 20%. So in people older than 60 or younger than 28. And for example, this peptide here is bound in 80% of elderly individuals, but only in about 10% of younger individuals. And the same thing we also did for sex, comparing males and females. So you see males and females are quite similar, but there are still some very significant differences between them. And then we also use machine learning algorithms to kind of take this information, predict age and sex. We can also predict inflammation markers such as CRP, C-reactive proteins, demonstrating that these antibody repertoires tell us a lot about human health. And then I always get the question, what's the point of doing this? Why do you do this elaborate immunoassay? Couldn't you just do regular microbiome sequencing? So what most labs these days are doing, they take stool samples, they sequence the DNA inside, and that tells you about the abundances of those bacteria. So that's relatively straightforward. That's easy to do. What we are doing, getting blood, making these phages, screening them, that's quite a lot of effort. So what's the added value? That's compared in these plots here. Here we kind of generated this metagenomic data and this antibody repertoire data for the same individuals. And then we compared what's, the predictive value of this metagenomic microbiome composition compared to the antibody repertoire. And you see that the antibody repertoire has a lot more information related to age or to sex than the microbiome composition. So it's not just, duplicating the information that you anyways get from the microbiome. It's much rather an additional layer of information. And why is that the case? Why does the antibody repertoire have a different amount of information? Well, I guess one key aspect is immunological memory. So basically, when you're exposed to something early in life, when you're vaccinated against something early in life, you will keep making antibodies against that for the rest of your life, ideally. So your immune repertoire really is an agglomeration of all the things that you've been exposed to. The microbiome, on the other hand, from the gut, is very much dependent on the time of sampling. Depending on what you're eating, your general health status, your microbiome composition is changing much faster. So it's more a snapshot rather than, this agglomeration that the immune repertoire represents. And this we have also tested systematically by comparing the immune repertoire of 200 individuals 5 years apart. So we collected samples at baseline and measured the antibody repertoire, and then samples of the same individuals 5 years later. And what you see here is all of those 200 individuals, and you compare, the first person at baseline with the first person 5 years later, with the second person, with the 100th person, and with the 200th person 5 years later. And the same thing you do with all individuals. So the 100th person with the first person, and so with themselves, and then with everyone else. And you can see that everybody correlates to the highest level with themselves. So you basically get a perfect separation between these time points. You can match 100% of these samples, like an immunological fingerprint. And if you would do the same thing with stool samples, there is also diagonal, but it's a lot more noisy. There's a lot more background noise, and you could only match about a third of these samples. So this really highlights this concept of immunological memory, that kind of our assay in a way allows to go back in time and see lasting effects of previous exposures. And this work kind of on this diversity and stability of these antibody repertoires has already been published. And then we had a couple of follow-up works where we have shown that these immune repertoires also play a role in different diseases. In inflammatory bowel disease, we can also study them in coronaviruses. And for example, in inflammatory bowel disease, this was done with colleagues in the Netherlands with Arno Bourgogne and Rinse Bersma's lab. There we had a cohort of Crohn's disease patients. And what you see in these plots is, again, every dot is a peptide. And then you have the percent of antibody responses in healthy individuals or in Crohn's patients. You can see many things are similar, but there's a distinct group of antigens that's massively overrepresented in Crohn's patients. And these are actually flagellin surface structures of certain gut microbiota. Interestingly, this is not the case in ulcerative colitis. Ulcerative colitis is also an inflammatory bowel disease, but is rather affecting the large intestines, whereas Crohn's disease is rather affecting the small intestines. And here you also see striking antibody repertoire differences, but that they are not targeting a single group. They are much more dispersed. And with data like that, we can also train our machine learning algorithms to basically accurately separate between these diseases and diagnosing them. However, this whole workflow is not limited to the gut microbiome. I mean, that's what we were mostly interested in, but basically you can use these synthetic DNA libraries to encode any types of protein antigens that you're interested in. So when the COVID-19 pandemic hit back in 2020, we made a library that covered all coronaviruses, so the current SARS-CoV-2, but also the original SARS from 2003, common cold coronaviruses, as well as animal coronaviruses. So we made 13,000 different variants where we could basically pinpoint single amino acid differences between these viruses. And we have then shown how there exist cross-reactivities between these viruses. And we can also, how we can use this for diagnostics. So I think what I've shown so far is more like obvious stories. So obviously there would be immune response in inflammatory bowel disease against the microbiome. This has been known. Obviously, there would be antibodies against coronaviruses. So we also try to apply this in a little bit unusual settings. So here we studied chronic fatigue syndrome, which is not long COVID or post-COVID syndrome. This is kind of started in 2017, the, let's say, original CFS, which is a debilitating fatigue, which has for a long time not really been recognized as a proper disease. And it has always been unclear, is it triggered by viruses? There was some data implicating the gut microbiome. So what we did here, we screened in an agnostic way. We just used our large antibody and antigen libraries and just tested the immune repertoires of these CFS patients and healthy controls. And interestingly, we found that they have elevated immune responses against a certain type of gut microbiota. And on the other hand, they're also lacking immune responses against different viruses, really showing that there is basically also a molecular signature associated with this disease. And we are also doing a little bit more basic research, for example, looking at links between genetics and immune repertoire. This is again with Dutch collaborators, where we have shown that your immune repertoire is shaped on the one hand by genetic factors. So there are basically 3 loci in the genome shaping your immune repertoire. But on the other hand, also co-housing, living with your family, living with your partner, also changes your immune repertoire simply by exposure to the same antigens. And maybe wrapping this up, one strength of our workflow is that we can combine all of these libraries. So we can measure the microbiota together with coronaviruses, together with other antigens. So we can currently measure more than 300,000 antigens in parallel. And that also includes allergens. So we also have a library where we encode different food proteins, environmental proteins. And we have also shown that we can use this to detect immune responses against food in healthy people and also in the setting of allergies. And maybe from the high level view, what is unique about our approach? What are other people in the field of systems immunology doing? Well, a lot of people these days are doing BCR and TCR-seq, which means you isolate immune cells, you isolate the DNA, and you sequence that DNA, focusing on, the B cell receptor encoding antigens or the T cell receptor. And what you get there is basically VDJ segments, mutations, clonality, diversity, what these repertoires look like. But the big problem is you don't really get a function. You don't know what they recognize. You just know the DNA sequences that encode the functionality, but you don't know what it binds. And in a way, I like to say that, in the universe, there's a lot of dark matter, and the same way in our immune system, there's also a lot of dark matter where we don't know the functional consequences. And that's the big strength of our approach, that we actually do get a functional readout. We do know the antigens that are bound, and we can study their role in different human diseases. And that's basically what the lab is doing at the moment. We're focusing on different cohorts, cancer, IBD, autoimmune diseases. And we're also planning to take this more towards animal models where you can study causality, as well as extending it on a technological framework to other directions, such as T cells. And as a part of that, we're always interested in collaborations. So if you're also a life scientist or a clinician and you have interesting cohorts where you think that these antibody repertoires are relevant, just reach out to me. I'm always very happy to discuss such ideas. And yeah, that being said, I would like to thank again Kais for the introduction, basically my lab for all the work, and also the funding agencies, primarily European Union, for supporting our research. And I don't know, do we have time for a discussion and question, or what's the framework?
Well, thank you, Thomas. And of course, we definitely do have time, and I think the others can think about it. But one thing I would already like to touch upon and kind of lead into the first question while others think about theirs is you mentioned at your start kind of your CV and the way you developed as a scientist and the way you realized, okay, bioinformatics are the next best thing. And I think also from my end, I can see that bioinformatics are playing a bigger and bigger role in research in general. In your opinion, is there even a possibility of just being like that traditional scientist in today's day and age where you're only known for wet lab stuff? Or do you have to be like proficient in the wet lab as well as dry lab analysis?
I think that's a good question. So I think it would be hard to completely avoid any type of bioinformatics. But the question is, do you really need to code yourself and you need to be able to run and write scripts yourself? Or is it enough if you can just use a pipeline that someone else has assembled for you and to just understand how to use it? So I think like completely avoiding bioinformatics will be hard. I mean, even if you're doing microscopy and you're heavily focusing on that, there's still nowadays a lot of imaging analysis that's done computationally, but there also exists software for that, right? So I guess you could probably avoid this whole bioinformatics, machine learning, data science aspects, but it very much depends on the field. Even in I guess basic molecular biology, a lot of people are doing single cell sequencing. That's almost becoming a standard method. And those data sets, even though like pipelines and workflows exist, they're so complex that they're hard to use without understanding a little bit of bioinformatics.
Thanks. But yeah, giving over to the others, are there any questions for Thomas? You can turn on your mic. You can also write it in the chat. I'll keep an eye on the chat as well.
If I may pose a question.
Sure.
Okay, thank you. Well, I'm a physicist. I'm not a biologist, so I don't have a background in this field. But you have mentioned in your last, some of your last slides, dark matter. So my question would be, can dark matter in your field, as you have phrased it, can be detected and tested? And if yes, how? What are the methods?
Oh yeah, sure. Maybe I'll share my screen again that everybody knows what we're talking about. So actually, like a bit of apologies from my side. So I didn't really quite know what the background of you guys was. So I thought you were more like life scientists, but it's very diverse here. So you're both okay. Well, then this slide was maybe a bit harder to understand. So it's like just an analogy that I was using. So what I meant is, in physics, you have, the universe, and there's a lot of dark matter where we don't know what it is, right? And in the human immune system, we have these cells that make specific molecules that bind other molecules, right? These are, like I said, for a coronavirus, that your immune system can recognize a coronavirus, it needs to bind something like a distinct structure on the coronavirus. And that's like one protein, for example. So for the coronavirus, there's a protein called spike proteins, and we know that humans make antibodies targeting that protein. But for the gut microbiome, there's not one virus, there's not one bacterium, there's thousands of bacteria that all have thousands of structures on their surface. So you can't easily test it. And that's what I call the dark matter. So I'm just saying, in the same way, the universe has dark matter. Also, you have all those microbes, and we don't know what our immune system is recognizing. And by this novel assay, this high throughput phage assay, this FIPSeq method, we can actually target this. So the key idea of my talk is that we have like a technology, this combination of phage display, next generation sequencing, where we can actually target this dark matter and find out what they recognize.
Thank you.
Sure.
And one thing I would also like to add, if there aren't any other questions before we move on, is I think you're kind of in the middle of every single possible scientific approach. And I think we also have some IT specialists within here. You're obviously working with FIPSeq, which produces huge data sets that are like obnoxious to work with. What are some of the biggest challenges of working with big data, and how do you tackle those challenges on a daily basis?
Mm-hmm. Well, I guess the key problem is the old garbage in, garbage out, right? I mean, if you have poor experimental data and you run, data science, machine learning on that, you will get poor outcomes. So you always have to be very much aware of any bias that your methods are introducing from the experimental side. And you always have to think what could be another explanation for the results. So, in the signals that we see, it's very easy and very tempting to say, okay, this comes from a disease, but it could also be that there's just some technical issue. For example, especially when you have a disease and when you have healthy controls, it could be that the patients have been, assembled in a hospital, but the healthy controls were assembled by general practitioners or something like that. And then maybe you just have some contaminations from the hospital or from the general practitioners. And what you think are differences between the disease and the controls, healthy controls, are not even caused by, real differences, but just in a kind of sampling bias, how the samples were collected. So I think that's the overwhelming problem. All of these problems related to data generation, especially for large human cohorts, are so much worse than anything you can do wrong on the data science side. I mean, there are obviously some mistakes you can make when interpreting that data. I guess I don't want to say doing it the wrong way, but you can use improper statistical tools. Also, these machine learning algorithms, let's just say you can sometimes just use the wrong algorithm, and that what you then get is not very meaningful. You have to be aware of those things. But the bigger problem, I would say, is getting clean data that's meaningful to interpret.
Thank you, Thomas.
Sorry, may I ask one question?
Sure.
If we have time. Yeah, I'm not sure 100% if I understood the technique properly, but you are working with microbiome, and I was wondering, can we apply the same way of analysis to other organs? For example, if we are working with, I don't know, brain or retina, for example, disease versus a healthy one, can we apply the similar approach of detecting epitopes or whatever?
Yeah. So I mean, I guess for the immune system, not so much, because like in the brain, that's behind the blood-brain barrier, and there's like almost no migration of microbes there. Also, like the eye is an immune privileged site, so there are antibody responses there. It's actually a good question what they bind. They are like also made by special lymph nodes, but in general, you would have a quite low load of bacteria. So there's not like a diverse microbiome where you have many different structures. It's more like a very limited set. So I think what would be more interesting, for example, would be the lung, because in the lung, you're also, constantly breathing, getting antigens in your lung. That would be a more interesting environment. Also, I guess oral immune responses because they are not the same as in the gut. They're also mucosal, but they are produced by different lymph nodes. And that's easier to sample, right? You can spit somewhere, and in your spit, you would have antibodies. The other aspect with the brain, it would also be very hard. The eye is very hard to sample. Nobody likes a needle in their eye. Usually people don't like that. And even like lung, getting samples from the lung, you would have to insert a metal tube to get like the sputum, the liquid out, which is also not very enjoyable. So what we are doing blood is mostly because it's easily accessible. But I agree that looking at spit, saliva would also be worthwhile.
Thank you.
Sure.
I mean, what do you have anything to add, or?
I think there's some feedback. We're not clearly hearing you, Amelia.
Yep, all right. But if there aren't any further questions, I would like to close it out with a final one, since I believe most of us are on their early research journey, and I believe some of us will eventually pursue a similar road to you, Thomas. And from knowing you personally, as well as being familiar with your work, I know you're leading the consortium on the Dark Matter project as of right now, which is huge and combining, I believe, 5 or 6 nations and about 15 different institutions. And you're always open to collaboration. So could you tell us from your experience, like the importance of such collaborations, but also the challenges of such collaborations? Because I believe as a coordinator of this whole consortium, I think you came across many different obstacles you had to handle and to tackle as well.
Yeah, so I think it depends very much on your field and the sort of research that you're doing, if you really need to collaborate. If you're a mathematician and you're solving equations, you can probably do that in your office. Or if you're like a programmer, a data scientist, you can also do it, I guess, on your own, but even then you would need some data to interpret, and you have to rely on other people. I mean, that's the main reason for me that I have a technology, but I'm not collecting samples myself. Like, I'm depending on other people for getting interesting diseases. I need clinicians who are collecting those patient samples, who send me blood, and then I can analyze that data. So for me, it really is a necessity for doing the work that I'm doing. And I guess with everything in life, it's hard to find good collaborators, because you have to find people who are motivated, who really want to collaborate, who are willing to put an effort, who are willing to share. Then you always have these arguments: how do you split credit, right? I mean, this is like in science with publishing, with the last author, with the first author. It's always a big debate. And even more strikingly, a lot of people are very easy to, show interest in the beginning, but when you then follow up and it's really the time to start working together, they are not responsive. So I would say I approach a lot of people and I discuss with a lot of people, but probably only with 5 or 10% the projects move forward and we are actually doing something, which is quite tough, because you have to put a lot more effort. You have to talk to a lot more people than you eventually end up collaborating with. And for the practical collaborations, if we have a consortium like the Dark Matter Consortium, there a big challenge is just the administrative issues. If you have blood samples collected in Spain and you want to send them to Vienna, it's not that easy. You need to get ethical approval that you can actually run these assays on these samples. And then you also need a legal material transfer agreement between the university in Spain and the university in Austria. Like, what happens if you find biomarkers? What happens if you develop a really powerful drug? Who gets a share of that? Who gets the patent? That's the most frustrating and I guess the most cumbersome part of it. So our project already started in January, and we are still in the process of sorting out all these legal aspects. But we knew this in advance, and we have basically assigned the whole year 2024 just for dealing with these legal obstacles. And we have 5 years in total, so we still have 4 more years from 2025 on actually do the science.
So I guess as a scientist, you're not just able to do science, but you have to wear the hat of an administrator and a law person as well. So
yeah, I mean, you know, for the sort of work that I'm doing, again, if you're a mathematician or even if you're like a basic molecular biologist who is just working on cell lines, there are a lot less issues associated with that. But whenever you work with human samples, which eventually, I guess, is the only way to really get medically relevant insights, then obviously, I mean, for good reasons. I mean, there has been a lot of controversy and a lot of malpractice in, you know, collecting patient samples in the past. So there is a reason for that, but still it's frustrating if you have to deal with all those legal aspects. I just see there is like one more person asking if we have time for another
yes,
question.
Selma, Thomas, if you're up for it, I
Sure.
would. Perfect. Selma, you can either write it in the chat or just turn your mic on and just let us know.
I'll just speak like this. Thank you, Thomas, for a very interesting talk. My question would be more of applicative nature. Do you know if the knowledge you get from these kind of analysis could eventually be applied for some kind of therapy development for patients with Crohn's disease, for example?
Mm-hmm. Yeah, that's part of this EU consortium that we are working on. So there are actually clinical trials in France for Crohn's disease, which revolve around you know, microbiota transfer. So the problem in Crohn's disease, like very much simplified, is that your body is overreacting towards harmless bacteria. So everybody has, you know, this gut microbiome, and Crohn's patients don't make immune responses against their own gut. They make immune responses against the bacteria that they have. So one simple idea would be that you just take microbes from healthy individuals and you transfer them to the Crohn's patient. So this is being done. The only problem is that, you know, everybody has vastly different gut microbiota. That's what I said with these thousands of different species, and everybody has a set of those. So what works in one person may not work in another person. And that's at the moment the challenge, finding out kind of in a personalized way, what is the right mixture of microbes that you should give a Crohn's patient. And there, as I said, we're only working on this for 10 years or so. So there we're just too far away from basic understanding to make it work. But there are cases where it works very well. We just don't understand why.
Okay. Then just another question that came up to my mind based on your reply, and it's just, is there something that's considered as a golden standard for the fecal transplant or that microbiome transplant? How do we decide, you know, which sample we want to then transplant to a patient?
Yeah, that's an excellent question. No, there is no gold standard. It's completely, I mean, it's not completely random. What is usually done is you select healthy individuals that are not obese, that don't have any viral infections, so they're screened for pathogens, and that's all. That you take that. In some settings, there's also, I didn't go into this cancer story, like there's for some immunotherapies, it is known that the microbiome has a beneficial influence. Some people respond to these therapies, other people don't. And in these cases, what they do is they take the samples from responders, people who respond to the treatment, and they give that to non-responders, and that actually works quite well. But in Crohn's disease, you can't really do that because there is no a response, non-response in Crohn's disease, right?
Very interesting. Thank you very much.
Sure. I see there was one more question in the chat. Someone asked if we could also to look at urine samples. I guess in principle, yes, there are, you know, bladder infections, and I mean, there's not a large bacterial load in the bladder, and I think the antibodies that end up in the bladder, they are just like filtered blood antibodies. So eventually, through your kidneys, some antibodies are not held back, and they end up in the urine. But it's an interesting question if there would be like a unique signature that you also have some mucosal antibodies there. Yeah, it's like also for me interesting because all of these basic things I'm so used, you know, to studying blood and, you know, the samples that are easily accessible, but it is probably worthwhile also trying these a bit unusual things. So thank you very much, Mila, for the suggestion.
Also, once again, from me, thank you, Thomas. I think we won't be holding you up anymore. I believe we'll be in Kiel next week. So happy travels. I hope that the first meetup goes well, and also good luck with the consortium as a whole. I hope to hear a lot of great things from it, and we'll let you go. If you'd like, you can obviously stay. We'll continue with our ceremonial part. But once again, thank you for taking the time. Thank you for the insightful presentation, and
yeah, also from my side, thank you very much, guys, for inviting me and having me. And like I said, it's always pleasurable to do these things, right? You can present your work, and like the discussions here were really interesting. I mean, I guess you guys, you're not exactly in my field, but still it's good to get a view from other people, and also seeing what did I explain well, what did I explain not so well, and what are things that you're thinking about if you hear this. So I very much appreciate it. Thank you for having me, and have a great rest of the ceremony, and have a great rest of the weekend, and see you.
appreciate it. Thanks. Bye.